Have you ever stopped to think that there's a hidden world of light all around us and even inside us? It's true. Our very own bodies are packed with molecules that can absorb and re-emit light, creating this faint, invisible glow. Today, we're going to dive into a technology that turns that tiny whisper of light into an absolute roar. I mean, really think about that for a second. What if we could find cavities, or even cancer, not with a painful needle or a biopsy, but just by shining a light and seeing what glows back? Well, this isn't science fiction. What you're seeing here, where diseased tissue literally lights up like a beacon, is possible thanks to a technique called laser-induced fluorescence, or LIF for short. It's an incredible way to make the invisible visible, and it's totally changing how we see our own health. So how on earth does it work? Okay, at its core, fluorescence is kind of like a glow-in-the-dark sticker. You know how you have to charge it up with a lamp before it can glow on its own in the dark? Well, in our bodies, we have tiny little molecules that do pretty much the exact same thing. The sciencey name for these molecules is fluorophores. But don't let that fool you. They're everywhere. Our tissues are just full of them. Things like collagen, the stuff that gives our skin its structure, and even certain vitamins. They all have this natural ability to glow. Just think of them as these tiny built-in light bulbs that we can work with. All right, this diagram, a Jablonski diagram, it might look a little complicated, but the idea behind it is actually super simple. So first, a molecule absorbs some energy from light. Let's say it's blue light. That's the blue arrow going up. This gets it all pumped up into an excited state. It kind of jiggles around for a split second and loses a tiny bit of that energy as heat. Then, to chill out, it releases the rest of the energy by spitting out a new particle of light. That's the green arrow. And here's the key. Because it lost some energy as heat, this new light is always a different, lower energy color, like green or red. That color changes everything. So, it all boils down to this simple three-step dance. Absorb high-energy light, relax for a moment, and then emit lower-energy light. That shift in color is the unique signature of fluorescence, and it's exactly what scientists are looking for. So, here's the thing. The natural glow from our body's fluorophores, it's usually way too faint to see. So to turn this into a tool we can actually use, scientists needed a way to supercharge the signal. And that, my friends, is where the laser and laser-induced fluorescence comes into play. Right, instead of using just any old light, we can hit the tissue with a very precise, powerful laser beam. This pumps a huge amount of energy into exactly the right fluorophores we want to look at, making them glow much brighter. This makes the signal so strong that we can spot even the tiniest changes in the tissue. But this brings up a really big challenge for the researchers. See, inside our bodies, molecules are all crowded together. Their glow can be messed with by all sorts of things. To really understand how a fluorophore works, you have to get it all by itself. And the solution is a device that looks like it's straight out of a sci-fi movie. This is a quadrupole ion trap. You can think of it as a molecular cage. It uses electric fields to hold a single charged molecule, or ion, just hovering in a perfect vacuum. It's basically the ultimate clean room for studying how a molecule and light interact with zero distractions. So the whole experiment goes like this. First, you turn your molecules into ions. Then you trap them. And you zap them with a laser, and some super sensitive detectors analyze the light they give off. It's this fundamental research that lets us understand the complex signals we see in actual human tissue later on. Okay, so we've got the basic science, we've got the research tools. Now, let's get to the real payoff. How is life actually being used in medicine right now? Well, it's leading to something amazing called an optical biopsy. This quote from the research really says it all. Instead of physically cutting out a piece of tissue and waiting days for a lab result, doctors can get instant feedback. It's painless, it's fast, and you can do it over and over. Let me give you a perfect example. We have all been there, right? The dentist poking around in your mouth with that sharp metal pick. Ugh. Well, life completely changes the game. Instead of that pick, a gentle beam of light can find tooth decay at its absolute earliest stage, way before that metal tool ever could. And, crucially, at a point where you can actually reverse it without any drilling. The sensitivity here is just incredible. Look at this number, 5%. Healthy tooth enamel glows really brightly under this light, but when it loses just 5% of its minerals, the very beginning of a cavity, that glow starts to dim. LIF systems can spot that tiny change. It means we're finally moving from a drill and fill world to a predict and prevent world of dentistry.
And it's a very similar story with cancer detection. This graph shows the natural glow, or autofluorescence, from tissue. The top two lines, that's healthy tissue, glowing nice and bright. The bottom two lines are from a tumor. See, cancer changes a cell's metabolism, and that changes its mix of natural fluorophores, causing its glow to dim way down. For a doctor, that difference is a clear, real-time signal.
And it's not just for diagnosis. LIF is also a critical partner in a cancer treatment called photodynamic therapy. Here's how it works. A patient gets a special drug that loves to hang out in cancer cells. That drug is also a fluorophore. So doctors use LIF to see exactly where the drug has built up. And then, bam, they hit that spot with another laser to activate the drug, which then kills only the cancer cells. It's brilliant. Now, as amazing as all this is, for any of it to work, the signals we're detecting have to be crystal clear. But sometimes those signals can be incredibly weak. So scientists are in this constant race to build better, more sensitive instruments to capture every single last particle of light. So on the left, you've got that standard ion trap we saw earlier. It works, but a lot of the light the ion gives off just gets blocked. But on the right is this brilliant redesign called the TAC trap. You see that? One of the electrodes is a giant curved mirror sitting right under the trapped ion. It acts like a little satellite dish for light, collecting photons from a much wider angle. And the difference this one little change makes is absolutely staggering. A standard trap might only collect about 2% of the light a molecule emits, but the TAC trap with that built-in mirror can capture up to 24%. That's a 12-fold increase in the signal. For studying molecules with a really faint glow, that is a total game changer. So why does pushing for this much sensitivity matter so much? Well, check out this weird mystery. This is about fluorescein. It's one of the brightest, most used fluorescent dyes in biology. In water, it glows like crazy. That's the dashed line. But when scientists put it in an ion trap all by itself, its glow just disappeared, vanished. The energy was going somewhere else. Understanding paradoxes like this is exactly why this fundamental research is so important. It helps us design even better tools for the future. So, from catching cavities before they even form, to guiding cancer treatments, to solving these deep molecular mysteries, laser-induced fluorescence is giving us a whole new way to see the world. It really makes you wonder, what other invisible secrets are just waiting out there, ready to be revealed by the language of light?